So I am the first scientist in my family. And it was interesting because that wasn't really something that struck me until later. Basically, I wanted to be a gene jock. I wanted to understand how to do molecular biology and pursue that.
The impression that Cathy made on me when she was undergrad, when I was a graduate student, was that she was very sophisticated as far as the way that she approached a research problem and willing to use any tool necessary to get to the answer of a particular question. She was starting to apply to graduate school, and I told her it doesn't matter as much what school you're at. It matters very much who you work with as a professor and what lab you're in.
The woman that I went to work with, Deborah Farber, she had identified the biochemical defect in one of the first mouse models of retinal degeneration that causes a disease known as retinitis pigmentosa. But we didn't know what the actual gene, which, the RD1 gene, this defect in the mouse, where it was coming from. So I set out to clone it.
This actually was quite a adventuresome undertaking. And at that time, I think only maybe 1 or 2 eye disease genes had been identified, period.
She gave me basically the freedom to do what I wanted. I came into a lab that did all biochemistry, and I wanted to do molecular biology. And there was nobody in the lab that did that. So she gave me the freedom to go to other labs, to learn how to do it, to bring it back. Literally, she didn't even have the equipment to do the kind of analysis that we did. And she was willing to invest in it as I was learning how to do the different things.
It was a risky project, and Cathy had to be somewhat brave to take it on. And Deborah had to be somewhat against her nurturing aspect to give somebody a project that could fail.
She provided an environment for me to grow independently. I had to come up with a strategy, a subtractive hybridization strategy that was just being developed. And it was a way to basically agnostically try to identify what the gene was without knowing what it was. And you overlaid them and you would look for differences. And I can remember seeing it. It was clone number 408. And it was really cool. And the rest literally is history. When I moved to Duke University, I had already transitioned into studying age-related macular degeneration.
Cathy made a shift. She applied some of the methods she developed and learned in this rare disease genotyping project on a mouse to try to find it in the patients.
We did not have anyone in research like Cathy, in any way like Cathy. And so it was exciting.
One of the things that I loved, was fascinating about her, is she said, I cannot wait until I don't have to do the surgery anymore.
People are coming at us with vision is falling away. And how do we stop it? What can I do?
You get this hub of these new ideas that can emerge and be either one direction looked at in the lab or in the other direction be tested in the patient.
We were both fighting from 2 different ends, addressing a similar challenge of macular degeneration.
We were looking at donor eyes. We wanted to figure out when we get a donor eye that has AMD.
Cathy's work didn't change that patient's outcome, but that's the person that she will affect in the future.
The clinicians and the researchers actually talk to each other. We interact frequently enough that these different collaborations can develop.
As it's an aging disease, the mice don't get it until they're halfway through their life. So the logistics of having enough mice and aging them for a year before you can start doing an experiment, many of my colleagues would have stopped before they worked on that problem.
The first mouse that we developed became a gold standard for really looking at how do you model AMD in a rodent. Biggest risk for AMD is age. So I wanted to incorporate aging.
She has them smoke cigarettes. She has them eat fat, So here they are on the worst of the American diet.
And then to add a gene that would have an impact. And the 3 of those, a multifactorial approach to developing these mice. Why this is important is that there's no other mice like that.
These are pieces of the puzzle that other people use. And they take this in and it informs their research. Or they use her mice to test their questions.
A number of years ago, the genetic risk association for AMD exploded. Literally, there were 4 papers that came out at the same time in 2005. One of the papers came out of Duke University, and one of the authors of the paper came to my room, knocked on the door, and said, "You need to make a mouse." We set out to make a gene therapy for AMD based on augmenting CFH. And what we can do in the mouse that basically de-risks it to get pharma interested in actually taking this all the way to humans is that because we have a model where we can actually show proof of efficacy. So if we can put the CFH into the RPE and prove that our mice no longer develop an AMD phenotype, that's the ultimate goal of what I'm in the middle of doing right now.
What my lab has worked on traditionally is delivery of genes to the retina to allow long-term treatment with a single injection.
What he has become is an expert in gene therapy.
So it was sort of a natural dovetail collaboration since we had known each other for decades, but Cathy and I had never collaborated to try to design intraocular gene therapy for the pathway that Cathy and Greg had discovered.
He has a grad student in his lab, Sonali, who's trying to develop an AAV vector that we can stick into the vitreous that will transduce the RPE cells. And that's my ticket to being able to targeting the RPE cell the safest, fastest way for patient use later.
She has an amazing skill at taking nascent scientists and moving them into the field.
These are the guys that they're in the trenches. These are the ones that are doing the work.
Energizing them and sharing her goals and focus.
Luckily, they run with stuff, just like I had the opportunity to do with my PhD.
I think Cathy's like a goddess. She's like the role model that I aspire to be. She's this amazing woman who's a scientist, who's a mom, and that's the person that I want to be in my future.
And that's more important now than ever because it's harder and harder to do this. A lot of these really bright minds, they don't want to become scientists anymore. A lot of them want to run off to pharma or not even do science at all. So it's really important and it's really inspiring to see.
I think Cathy rescued me from potentially not pursuing science anymore. And I don't have a lot more words to put on what that makes me feel like.
She's one of the people that's kick-started, that's moved people, that has really moved the field forward and not just taken part with everyone else and doing something. She's really leading the way and moving us into new directions.
The quality of life for AMD patients is pretty awful. It's not so much that it's painful, it's that it's isolating.
The disease was impossible to accommodate because macular degeneration, what's that? Oh, okay, you are old. You are old and it's okay not to see.
She was burning herself at the stove trying to find out whether or not what she had in a pot was hot.
They lose their mobility, the most important part of their vision, their ability to recognize their family's faces, to read.
My concern was about her physical safety, but it was also about her ability to do things other than just sit by herself and be alone. And then she started losing interest and started becoming more introverted.
So she started closing the shutters because the light, bright light, will hurt her. You will go into her apartment and you will see her with the shutters closed, the bright. She will live in the dark. Oh, that made me feel like she was in prison. That made me feel so.
I support medical research entirely. I'm not necessarily programmed to be the guinea pig, but if somebody says, here's your condition, here is your prognosis, and this is what we might be able to do to help you, then I'd seriously consider that.
How do we get to do the grassroots it's going to take to get people to pay attention to this disease and to raise more funding, to do more research, and to develop therapies? And importantly, to make sure that pharma also still stays interested. You would think it'd be a huge market. It is.
That's one of the reasons that I'm always telling people, she's someone that you should support with research money. If you've got money to donate, give it to her. She's going to put it to great use and it's going to change the world.
My long-term hope, which I hope doesn't take very long, is to see this therapy get into humans, to have an impact on the quality of life and the vision of patients with AMD.
I think it's really cool. Honestly, I love that she's gotten this award, and I love that it points out to Cathy how much people are valuing and recognize what she's doing. What better award than giving her a video so that she can share that with others and continue to, that we amplify what she does so well.
AMD affects over 200 million people, and having interest of everybody on this disease is what it's going to take to get rid of it.